who has agreed to come and give a talk at this workshop. She's going to talk about RNA branching. And so Christine is a combinatorialist who's been working in biology for a very long time, longer than me. I first met her when I was a grad student and thinking about making this transition into neuroscience. And I was advised, like, oh, talk to Christine. She's done something similar. And we've known each other ever since. And, you know, it's been a pleasure to, you know, follow her career and introduce her now.
Thank you so much. And thank you for the invitation. I'm delighted to be here. As you can see, we're going to be talking about a different biological problem. And so I've made the decision to suppress as much as possible the biological details and just try to focus on the math. But at the end, I'll get back to what a biological application would look like. So as Karina alluded, I work in actually Bernd Sternfeld at some point told me I should start calling discrete mathematical biology. It's like, if you do discrete math and mathematical biology, you should just say you work in discrete mathematical biology. That's great. So I do like to think about, very much like to think about combinatorial problems. And so a combinatorics person will often demonstrate that they've understood a problem by being able to count instances of things. And so here are two well-known, here's a very well-known sequence of numbers that's going to come up in the talk. And here is a less well-known one that we're also going to get to. Biologists so often will demonstrate that they've understood a problem by performing experiments. So they generate a hypothesis, they test the hypothesis, and so this is how biologists demonstrate that they've understood something. And I'm very interested in the interaction between these two things, and this is where I have to walk over and hit forward. And to make the connection a little bit more transparent for this talk, what we're really thinking about trees and how branch structures, trees, can be encoded in strings. So we haven't gotten to the strings yet, but this is what an RNA structure might look like more abstractly. And this is its corresponding branched structure. And so combinatorics people like to think about branch structures a lot, and molecular biologists who touch on RNA think about these kind of secondary structures for RNA. Now, you might be asking yourself, why do we care about RNA? In which case you don't remember COVID, for instance. So, you know, most viruses code their genomes in RNA. And so that's an important thing. We're able to be in this room, you know, freely because of mRNA vaccines. And also RNA is implicated in a lot of gene editing and regulation. And so again, a theory about what differentiates us from corn is not the number of our genes, but the complexity with which the genes are regulated. So RNA is important on many, many levels. We're going to be primarily thinking about short RNA molecules today because they're much easier to visualize. So here is an example of what is known as a tRNA molecule. So, and it has a structure that exists in 3 dimensions. So what, you know, in 3 dimensions, we've got the overall conformation of the molecule, and we care about this, we care about the structure because we're trying to infer function. And so biology, molecular biology, much like architecture, function follows form. So you really need to understand, you know, a way of trying to infer what the molecule does is if you know what the structure is. So however, understanding RNA structure at a 3-dimensional level is still a very difficult problem, and AI is not going to solve it anytime soon because most RNAs don't crystallize well. And so we have very little training data for which we can try to understand structure at the 3-dimensional level. The understanding structure at the 1D level is a solved problem by now. It is, you know, you can just basically sequence anything. In fact, they don't do sequencing in the experimental labs anymore. You just, you know, send it, it's something you mail order, you know. So this is solved, this is still hard. And the intermediate step is what is known as a 2D structure, or the secondary structure. And that is the set of base pairs, the intra-sequence base pairs that this structure forms. And the important thing that for the purposes of this talk is to understand that when I have stacked base pairs that are represented by these colored rectangles, that is thermodynamically favorable. So we want to maximize the number of stacked base pairs. And the single-stranded regions or the loops are understood to be thermodynamically destabilizing. And so we want to try to minimize the loop costs. So we have this fundamental optimization problem of maximize the stacked base pairs while minimizing the loop costs. So now, one question that you can ask is, well, if instead of, so oftentimes we'll think of, I'm given a sequence and I want to predict what the secondary structure is. But you can also think, I want a secondary structure. I'd like to generate a sequence that folds to that particular structure. So here is an example. So here is a structure much like the one we were just looking at. So it's a tRNA structure. And you can think about, I'm going to sort of abstract this structure to its graphical skeleton. So I'm going to put a vertex where each of the loops are, and I'm going to have those loops connected by edges, which are the stacked base pairs. And so I have now this skeleton of the secondary structure. And then if I want to design a sequence that can fold to that, I'm going to do that very simply. I'm going to put GC base pairs because they are thermodynamically the most stable. So I'm going to put those along the edges, and I'm going to put A's because A does not interact with G or C along the loops. And I'm going to say, ta-da, because I have created a sequence that will fold to my secondary structure. The problem is can does not mean does. So, the sequence that we've created here can fold to the structure that we wanted, but what its minimum free energy structure is a different one. So the sequence exploits symmetry to find a structure that has a slightly lower free energy than it would have if it folded to this one. And here, this table just gives the different trade-offs in the labeled loops. And so it is the difference in free energy is entirely due to the differences in the loops because it has the same number and type of base pairs. And so this was a problem this was the first RNA structure problem that I worked on was, is it possible to design RNA, or how difficult is it to design RNA sequences? And because at the time I was actually a postdoc in the computer science department at the University of British Columbia working with Ann Condon, who was there, and also Holger Hoos, we ended up doing a very computer sciencey thing, which is to say, we're going to take this hard problem that is designing secondary structures, and we're going to reduce it to another hard problem that we don't know how to solve. And so we're going to say, if we could figure out, if we'll give an explicit reduction where we will say, we can design the secondary structure provided that we can generate sort of we can solve this word problem where we have different enough encodings with which to put on the helices. And so this one sort of loop helix trade-off, actually, then I've been mining for the next 2 decades in terms of the research questions. So one that I'd like to come back to is the question of this loop energy trade-offs. And this is something that I think we're now sort of 9 papers deep into a collaboration with a former postdoc who's now a full professor at Clemson, Svetlana Postanovic. But today I also, because of the more mathematical nature of this audience and also the difference in the biological problems, I wanted to primarily tell you about the helix exchange combinatorics. So here we are thinking about, so here's an example. I've taken a RNA sequence here and I've made a very combinatorial one. So it's much like the design sequence. So I have A's here, and those are intended to be my loop regions. And I've got G's and I have C's. And so the run of G's can pair up with runs of C's, but they can do it in many different ways. So they are not obligated to pair up in ways, the first run of G's is not obligated to pair up with the first run of C's. The first run of G's could pair up with the last run of C's, for instance. And so for this sequence, and then I take this string and I copy it 3 times. And so when I do that, there are 5 different secondary structures that I can get that have the maximum number of base pairs possible. And so those are the ones that are listed here. And I'm using this arc diagram notation to simplify the matching. So this structure here, the first run of G's paired with the first run of C's. So that's why I have a 1 to a 2, for instance. And the second run of G's paired with the third run of C's, and that's why I have a 3 to a 6. And then I can think about, given this configuration space, how can I move around it? And I can do that by this helix exchange operation, where if I want to move from the structure that I was at here, for instance, down to this one, then what I'm doing is I'm breaking apart these two helices and I'm repairing them in another way to get to this particular structure. Great. You're moving the microphone, yes.
No, I can't see the pointer very well.
Okay, so I will switch to using the stick then.
Okay.
Thank you.
Yeah, it seemed very important for this.
All right, so this structure here, actually well, yeah, that's fine, I'm tall enough. This structure here has that I've labeled the left and right sides of the helices. And then this, what is called a non-crossing perfect matching, if you care or recognize it, is I've just taken those indices, and then I'm using the arcs to indicate the ones that have paired up with each other. And so for this particular sequence, when n is 3, so I've taken this string, 3 copies of it, I have 5 different secondary structures that have the maximum number of base pairs, so are saturated. And then I can think about the relationship among these 5 possible secondary structures. And the relationship is defined by this breaking apart and repairing operation. So I could move from this one to this one by just breaking apart the base pairs between 3 and 6 and 4 and 5, and instead pairing 3 with 4 and 5 with 6. Right? So this breaking apart and repairing operation then gives us some combinatorial objects that we're interested in understanding much better. That's going to be the purpose of the talk today. But instead of drawing secondary structures, we're going to move to thinking about what I was called their graphical skeleton. So this is an object that is known as a plane tree. That's what Stanley calls it. But if you go searching through the literature, you might also find them known as linear trees or ordered trees. So it's always, a little bit challenging when we can't agree on exactly what the name is. But it is a rooted tree whose subtrees are linearly ordered. So if I have 4 vertices, as unrooted trees, I have 2 distinct ones. I either have the star or I have the line. If I then choose a root, a unique identified vertex, then I have 4 trees. Because when I have to choose a root for the star, if I choose the one that has 3 children, then that differentiates it from the one that has 1 child and then 2 more. In the line, so I get 2 of them, because I either choose a leaf as the root, I choose something that has 1 incident edge as the root, or I choose something that has 2 incident edges. And then the ordering distinguishes these 2, because this root knows that its right child this root knows that it has a grandchild on the right and not on the left, and then left versus different for this one. Okay, so these are plane trees. And they are one of the very many, hundreds and counting of combinatorial objects that are enumerated by the Catalan numbers. So this is a really well-studied sequence of numbers. And then what we're going to do is we're going to label the boundary of our trees in a pre-order walk. So you could just think about it as I walk around the boundary of my tree and label it with the indices from 1 up to 2n. And then I'm going to denote an edge as the pairing of index I with index j. And I'm going to observe that in the tree, then I'm going to call an edge odd if the first index is odd, and I'm going to call it even otherwise. So I have then a parity to the edges. And we can observe that the children of the root always are odd edges. And then it alternates odd with even. And so you have this flipping of parity as you go through the tree. And our breaking apart and repairing operation becomes a well-defined pairing exchange on the edges then. So here I've drawn my edges as fat rectangles, and I've colored each half of them so you can kind of track what happens to it. So a pairing exchange consists of breaking apart the two edges that I have into the 4 constituent half edges, and then repairing them in the opposite way. Now, the really important thing here is that this preserves the edges, but it does not preserve the vertices. So you want to think that on each of these 4 sort of parts of a vertex, I have subtrees hanging off. And so this operation is really pushing or pulling, it can pull apart. In this one, these 2 subtrees were farther apart, and then they get pulled apart and pushed together so that they end up being adjacent to each other on this one. So it really changes what the biologists call the topology of a tree. And every time I'm like, it just makes me shiver. But it really, it really changes how the tree looks, basically. And so we'll see a picture in just one sec. Okay, so here, for instance, this is often so here's an example of it. To move from this one to that one, I take these 2, I pull them apart, and then they become these 2 edges. And this one sort of gets sucked up to be incident on the same vertex. So when you have this new combinatorial object, then the first question you ask is just, is my space connected under this operation? And you can see fairly easily that it is, because you're just going to look at an induction where you say, I either have, if my root has more than one child, then it decomposes into 2 subtrees, and so I can inductively then those are connected. And if the root has 1 child, then I'm going to operate on that child and its first child to get something that has exactly this. Okay, so those represent then, so here are the dashed lines here represent all the local moves possible and all the pairing exchanges possible in this graph. So we know the graph is connected, but then we are going to, and essentially this proof says they all, every tree is connected to this one, where it's the star with the root has the highest degree possible. I obviously have a dual one where everyone is connected to that one instead. And yeah.
Would you mind showing like the bottom one and the right one? How do you go
this one to that one?
Yeah.
Absolutely. So this edge stays fixed, and these ones, I take them, they're like this, I open them up, and then I do that. So it really is this, I do this. Exactly. It is exactly like the fortune teller kind of thing. And so you're just doing this kind of flip operation on them. And it's like, and with the trees, it can be very hard. And if you don't kind of match up this, if you don't do the matching between, okay, this edge stays the same. And so these are the 2 ones. And as the size of the trees grows, then it can become increasingly hard to figure out, okay, where exactly, what changed here. But back to this question of barrier. So any 2 trees are connected through a star tree, but I'm claiming that that's a high barrier path. Well, where did the barrier come into this? The idea is these trees are supposed to be somehow a fairly abstract model of our secondary structures, and loops with really high degree should be thermodynamically very unfavorable. And so what I'm saying is I can transition from any secondary structure that has n helices to another secondary structure with n helices, provided my sequence is sort of combinatorial enough. But in order to do that, I'm going to have to pass through a configuration that is thermodynamically very unfeasible. And so, so the question is what high barrier is that? Does that mean low energy or high energy? It means high energy. All right, so here's the kind of picture, here's a landscape picture. And basically what we're saying is, here we want to think of blue is low energy, right? Red is high energy. And the idea is that if I want to transition from sort of a configuration here to a configuration there, I'm going to have to kind of go over this mountain range. And that makes it very infeasible that that would happen naturally or without any sort of change to the system or assistance. So this barrier height problem is a well-known problem. And it basically is just answering the question of what is the cost of transitioning between configurations. And then there are obviously many variations on this. And so what I've somewhat facetiously said is all non-propositions. So, you can ask how am I measuring the cost? What does I mean, what are the steps that I'm allowed to take in my transition? And what exactly do I mean by configuration space? And so here are some references if you're interested in following up more on that. For us, though, we're going to think about it. Our cost is, so our configuration space is going to be the set of plane trees with n edges. Our step is going to be one of these pairing exchange moves. And so all we need to do is to define what we mean by the cost. And the cost is actually going to be, will end up being a function of the branching degree. And the justification for that is this idea of we can assign weights to the vertices in a way that is consistent with the thermodynamic model that the biologists use all the time in their optimizations. And so here, what we've seen, these are the 5 trees with 4 vertices. They're now rooted at the bottom, and that's why this vertex is open rather than at the top. And then these are the scoring functions that we have. So if I have a leaf, that gets this score. If I have a non-branching one, it gets this score. If I have a branching one, it gets this one. And if it's an external one, it gets that one. Catherine? I can repeat the question.
What's the significance of it being rooted?
So the question is, what is the significance of it being rooted? It's rooted because we have the ends of the sequence. And so, and we want to keep track of where the end, and also because the sequence has a left to right ordering, and that is also why it's not only rooted but ordered, is because we are limiting the number of rearrangements that we're going to consider equivalent secondary structures.
Thank you.
Okay, so here's the math problem that we're going to be considering is I want to characterize sort of low barrier paths in these graphs. So again, my vertices in the graphs are trees with n edges. The edges in the graphs are one of these local exchange, one of these pairing exchange operations. And so those are represented by the dashed lines. And then my cost function is going to be what I'm calling branching skew here. Okay, so the C of t is the number of odd edges in a tree, and it increases from left to right. So this tree has 1 odd edge, this tree has n odd edges, and as I move from left to right, a pairing exchange increases the number of odd edges by exactly 1. So I know that these trees have 2, those trees have 3, and that one has 4. Nikki? So the question was, what's an odd edge? So, and recall that the edges in the tree are composed of a left index and a right index. And when the left index is odd, then it's an odd edge. Yep. Correct. Yes. In the line it alternates. So, and we'll get to a picture again, and I'll show you indices again in just a second. But the reason that we're using the absolute value of this is because the trees in the middle will have the most balanced between even and odd edges. And that means that they also then, and that by extension means I'm trying to avoid the word average, typical, and anything that implies this, but sort of in general, they then have the lowest degree of branching. So the 2 star trees have the maximum degree of branching, and the middle ranks have the lowest degree of branching. And so what I'm really measuring with this is how close to the middle am I? So the farther that I am from the middle, the larger this value is going to be. And then that has implications for what the maximum degree of branching in the tree is going to be. Okay, and then we're going to answer 4 different types of questions. So we're going to start by thinking, I'd like to first characterize when there is a path that moves only left to right. And here, left is always going to be the star tree that has 1 odd edge, and right will always be the star tree that has n odd edges. So I'm always moving increasing number of odd edges. Then I'm going to answer the question of, well, I'm allowed to reverse direction exactly once. So I can go up to, I can move right to a certain point. So I usually think about up and down, but on slides it ends up being left and right. So I can move right to a certain point, and then I have to reverse myself and move left, or vice versa. So those are the V paths. We're also going to characterize that are 1 away from optimal. So the lowest barrier height is always going to be, you can't do sort of any better than where you started and ended. And the 1 away is because if I start and end that I'm the same rank, then I have to allow myself at least 1. If I start and end 1 rank apart, I might still need to go a little bit below or to the left or the right in order to be able to do it. Otherwise, it's going to be tight. And then we'll also think about the length of these paths, because we can easily see that there is a nice bound on what the length will be if I don't care about the branching skew. And so then the question is, well, if I do care about branching, how close can I get? Okay, so here is the most important part. All right, so we have 4 trees here, and I've given the labeling on them, and the odd indices are now highlighted in blue. Okay. And I'm going to, and what I'm going to do is define a tree partition of the odd indices by looking at so a tree partition of, I realize there's no definition because it's just a lot easier to show you pictures and hope you can follow. So the tree partition of T with the star tree with 1 edge, what I do is I take T and I write it on top in its arc form. And I take L4 and I write it on the bottom in L form. And then I follow the closed loops. Okay, so the first closed loop has the 1 in it, so that's where I get this one. The second one has the 3 and the 7. And the third one has 5. Okay? So then I can think about T with the other star tree, and I get a different partition of the odd integers by following the closed loops. Okay? So these 2 are straightforward.
Christine, is it obvious that the even integers you don't have to track separately?
It's an interesting question of if you swap odd and even, like what happens, if I exchange odd and even in the definition of this, I get different partitions, and how do they relate to each other? We'll get to that. And it's not that we don't track the odd indices at all, the even indices at all. This one explains how we have to track the even indices, okay? So T with T prime, okay? T prime has these arcs, okay, and so I get 2 closed loops, but I've said the partition, I've taken the union of them. And the reason for that is because what I start with the simply aligned subsets, and then I have to ask what happens to the even edges. And the even edges are required to still be a connected subtree. So in the previous 2 examples, because the stars basically did not trigger this constraint in any way. In this one, what happens is, so I look at the first loop and I say, okay, 1 and 5 need to be in the same subset together. And in this tree, the edges with indices 1 and 5 are connected, but in T, they're not connected. And so I have to take the union of the 1, 5 subset with something that then makes the minimal one, which allows me to have it be connected in this tree. So this is how the even indices come into play as well. So I need to have my partition that I'm getting I start with these closed loops, and then I take the smallest union of the closed loops such that the induced subtree, such that I get induced subtrees in both of the trees that I started with. All right. So once we have this definition, then we can actually start characterizing what the paths look like. So I move from left to right. I'm going to move from T to T prime, left to right, provided that T has a min-max decomposition with T prime. And so what that means is that when I look at their tree partition, then, so they're, often one direction is easy. So the easy direction is if I do pairing exchanges on disjoint subsets, don't cause any problem. So the harder direction is, well, I'm going to take this, tree partition, and I'm going to say, what happens when I do one of these pairing exchange operations? And what happens is that they are basically merging they're merging those closed loops in a very predictable way that you can characterize and then show this preserves this min-max property. And so the min-max property is that I look at the tree partition and the induced subtrees have to be each a star. So we can go back to, for instance, so first of all, let me see. This is T, this is what T looks like with one extra edge from the previous slide. And then T prime, I put another edge here, okay? And so they are not above each other. And so here, the induced subtrees here, so the induced subtrees in this one are all stars. So one edge is a star. This is two edges with root, yes, and this is two edges with the line. And this is one edge again, okay? These ones here, the induced subtree from the tree partition is not a star because this is my tree partition and I get a line and this one instead. So these two trees there's not a direct path between them in the graph, which moves only left to right. Yeah.
Sorry, I didn't understand. Why wasn't that one why didn't it satisfy the property?
This one?
Yeah.
Okay, here, because these are just the closed loops. This is actually the partition. And so because I have to take the union of the sets. And so this partition is the whole thing. And so that means that the induced subtree is the whole tree, and this tree is not a star. And that tree is not a star. Whereas for L4, any subset that I pick is going to give me a star. And then for T, the first edge is a star. The last edge is a star. And this one is the star on two edges that's at the bottom. Okay, so we can characterize what the trees look like when we're moving from left to right. And so then the paths where we're allowed to change direction exactly once we just basically build the apex out of the tree partition. And so we start with, it has to be above both of these. I find the tree partition between the two of them and do it. So let me just move to the next slide because I'll have trees back. Okay. So here are the two trees that we were talking about in our example. The line lives here, this one lives here. Okay, and so if I'm allowed, so they're the lower V path is this one, but then the upper V path has to go through this star tree and down to the other one. Okay. So if we look at these graphs, if we just think about these as graphs, they actually have a lot of really nice combinatorial properties. They're n-partite, as we've been observing. You can show that the diameter is n-1. The disjoint sets are enumerated by the Narayana numbers. And there is basically exactly one combinatorial object that has all of these properties. And it is the lattice of non-crossing partitions. And so what if I think about just all of the paths from this tree to trees in here to any other tree, that is going to give me that is then gives me an isomorphism with the lattice of non-crossing partitions. So if you're not, so a partition of, so we're talking about set partitions here. So now I'm thinking about the integers from 1 up to n. And it's non-crossing if when I draw the convex hulls of the partitions on a circle, they don't cross. Okay, so here's an example. That's a non-crossing partition. This is a non-crossing partition. But if my partition had 1, 3, and 2, 4 in it, then it would be crossing. And so that's not a partition. That is a set partition of the integers up to 4, but it does not then satisfy a non-crossing partition. And so our tree partitions, if we take the tree partition with L4, that gives me this one, and then I just project down from the odd integers down to all integers on, and that gives me exactly these, exactly a non-crossing partition. And then here's an example of another one, and it is related to the previous non-crossing partition in an important way, which is that if you have in this lattice, you want to know when are two of the objects, when is their least upper bound and greatest lower bound the top and the bottom element in our lattice? And so their lattice was originally defined by Creveros in 1972, and he said, here is a operator that given a non-crossing partition, I can generate one that is its lattice complement. However, and this is not an involution, so you do actually get cycles in the lattice, but it's a somewhat complicated definition in the sense of you take your original partition, you write a second copy of the integers on the same circle, and you draw the largest non-crossing partition that you can given the constraints from your original one. If you think about it in terms of trees, all you end up doing is you're just rerouting the tree. So rather than having this interleaving between two different partitions, you actually have a single tree and you're moving the root around the tree. And that is exactly the Creveros complementation operator. And so that if you recognize then that these are ordered unrooted trees, you can then count them, so the number of orbits under Creveros complementation, using either standard tree counting methods, so Perry, Prinz, and Tutt, Walker-up, Lebon, and I can never remember their names. Bergeron, Lebel, and or by the cyclic sieving phenomenon. I know, terrible French accent. Or by this, and this morning I was like, oh, I really need to be able to rattle off their names correctly, but alas. Or by the cyclic sieving phenomenon from Ryder, Stanlett, and White at Minnesota. Okay, so that's one. So that's nice in the sense of there was this open counting problem in a very well-studied combinatorial object that gets resolved, motivated by a question in biology where you're thinking about folding and unfolding RNA sequences. The next question is still open in the sense of you can think about, so the two trees are complements of each other in the lattice. Exactly when I draw I do this tree partition and I get a single closed loop. Okay. So you can ask yourself, how often does that happen? And so what we're looking at here are, I fix the bottom set of arcs, and then the top arcs are the ones that will give me a single closed loop with those. And again, I have a local exchange operation here that allows me to connect them. This is a two pairing exchanges that I'm making on the top to allow them to be connected. But I get this graphical structure here that has, so looks interesting. And then these are corresponding graphs where the bottom now has 5 arcs, and these really bug me because they have a lot of symmetry to them, and some of the symmetries can be explained and some of them I have not yet managed to explain. So for instance, this one has two symmetries of degree 2, and that I could flip top and bottom and left and right. And I can see there's one symmetry of degree 2 that I can easily see on the bottom, because if I move these arcs over here, then that's clearly a left and right symmetry. Not exactly sure where the second one comes from. And it took quite a while to figure out where the symmetry of degree 3 comes from, and that you can rotate it, and there's a symmetry of degree 3. So there's, you can fairly quickly get to long-standing open enumeration questions from this, thinking about RNA secondary structures, reconfiguring. But back to our barrier heights. So that was all just thinking about, moving in one direction. Now we're going to allow ourselves to zigzag as much as possible, but we're going to try and keep the branching degree as, as narrowly bounded as we can. And so we can do that really literally by just zigzagging. So I had 2 undergraduates that worked with me, and they were the ones who, figured this out at first. And so essentially, if I start on the same rank, then I need to be able to move 1 away. As I said, I can't stay in the same, I have to be able to zig a little bit. If I start 2 away, I may need to drop down, but otherwise, if I'm more than 2 ranks away, then I can always move between them in a reasonable way. But the length grows rapidly when you're doing this. And so it's, an interesting question might be, can I move between them as efficiently as possible in terms of the branching skew and the path length? And we can do that. A sufficient condition is that these closed loop decomposition has what I call the block decomposition, meaning if every closed loop is actually a subtree, then you can operate just on those subtrees, and you will have, you can have a bounded skew path that has the right number of edges to it. What happens if you don't have that is still still open.
Christine,
Mhm.
do you think in the biological setting that this is also happening, that you're sort of that the RNA structure is sort of reconfiguring itself with some probability?
Yes. So I think, Karina has asked, very politely, so what the heck does this tell you about the biology? And the question is, well, so one of the things in the previous proofs, you actually start every reconfiguration with sort of the leaves in the tree. And then their question is, and it is known that the hairpin structures are a very important aspect of RNA secondary structure. And I actually had this even before I knew that I had found this connection to Huntington's, for a different math bio workshop. It's not here just because this is a neuroscience one. So, there is this potential. If we understand better the role of hairpins in RNA secondary structures, that might well tell us important things about the biology. There is also this, here was another example of, these are reconfigurations that they found in influenza infections, and that those also seem to be suggestive of it. So it is a very, we moved pretty far away from the biology, but then you can look at that and say, so what insights might we glean from this much more theoretical analysis? And it does suggest that the hairpins are important in terms of how much kind of structure we reconfigure, and also that that sort of local reconfigurations may also be critical to that. So that was the, it's not entirely a spherical cow, maybe it's got a few legs and arms to it. I'm not completely sure. But I do care very much about actual biology and improving predictions. And so I wanted to just highlight like another branching problem question, and that is, if you really wanted to improve prediction accuracy. So here's an example again of a tRNA sequence, and this is the MFE configuration, and it looks basically like our design sequence did. We're supposed to get a four-armed cloverleaf structure, but instead this sequence, which is a real tRNA sequence, the thermodynamic optimization says its optimum would actually be this three-armed one instead. And so this sort of suggests that we can perhaps improve prediction accuracy if we understand better how the thermodynamic model is scoring those branching loops. And so so this is the part that I've been working on with Svetlana. Well, it started with another postdoc, Valerie Hauer, actually with my colleague, Yuri Bakhtin, first of all, and then with postdoc Valerie Hauer, who was the one where we looked at the tree space, where we're thinking about what are the optimal configurations where I'm allowed to arbitrarily vary the branching parameters. And so here we're dealing with trees instead of with secondary structures, but what I can see is that there are 4 possible optimum depending on the trade-offs in my branching parameters. And so this one, branching is not favored, and so the optimum is a line. These 2, branching is so highly favored that the optimum would be a star. And then the one which is most like the current parameters that are actually used, we have a sort of, we're branching kind of as much as possible, but keeping the degree of branching to a minimum. And then we started looking at actually what happens with using this idea of convex polytopes and their normal fans to look at real configuration spaces. And so in just the last 2 minutes, I'm going to blitz through these slides. So the one take-home point from this slide is that I can formulate the branching parameters as a linear program. This comes directly from the thermodynamic model equation on the top. It has 3 real parameters, A, B, and C, and then this dummy parameter that I put in to make it a linear program. But all of our work has suggested that, and the biologists generally agree, is that maybe the B is not important. So we've really been focusing on the A and C trade-offs. And so A is the initiation penalty, and C is the penalty for sort of the complexity, which is the number of branches in the loop. And then if you're not familiar with this concept, the idea is I have a linear program, and so it's defined by a normal vector to the hyperplane, and I'm thinking about optimizing that over a convex body. And so the optimum is always going to occur on the boundary of the convex body, and there is a duality, literally a one-to-one correspondence between an optimum and between the facets of your boundary and then the cones in the normal fan. So if you want to understand the normal fan, which is the parameter decomposition, then you can understand the convex body that you're optimizing over and vice versa. The reason that this is important is because we didn't currently know either, but computing the convex hull seemed much more tractable. So there is an algorithm originally due to Grimbaum and then implemented by Peter Huggins in his PhD thesis that is optimal in the sense of the number of calls to your thermodynamic optimization, so the number of calls to your oracle, you're making exactly the number to discover each vertex and each face of your polytope. So we actually, I heard someone, I think it was Catherine, mention a mathematics research community. So Svetlana and I were participating, led a project at one of those. Karina was actually there too. And so we looked at, and so these are slices, for two-dimensional slices of the four-dimensional normal fan. So we're only looking at b equals 0 and d equals 1, computed from our polytopes. And the important point from this slide, well, two actually. One, time is a factor here. So for tRNA, when we had 76 nucleotides, it was already taking hours, to compute it. When we moved up to 120 nucleotides, now we were at days. We actually tried at some point to compute about 300. That took us nucleotide things. That took us to months. So it can be done provided you're willing to wait long enough. So we started with, we don't have many polytopes, but when we look at them, we see this sort of recurrent pattern. And then I say, you have these math conversations that seem insane, like why is it a broomstick versus a bow tie? And then at some time it's eventually you realize that a broomstick is a degenerate version of a bow tie, and you can prove this exactly. So we have a full characterization of the infinite parameter regions. And this is the point where I say, so there's no biology whatsoever in this slide, because our characterization applies to any RNA sequence at all. But if we zoom in to sort of the finite regions, that's when we get back to the biology, and we can ask ourselves, can we improve the prediction accuracy? Because the current parameters, which are represented by this dot here, can be quite close to the boundaries of things. And so in this case, this particular sequence has a very good prediction accuracy because the dot is lying in the purple triangle. But if it were a little bit, if this were wiggled slightly, it might end up in this pinkish region or this bluish one, and then the accuracy might change substantially. So we have multiple papers basically investigating this. And I'm not expecting you to unpack this slide at all, but what we were looking at was how much can we improve the prediction accuracy if we have these slices from our particular test sequences. And so we have two kinds of test sequences. We have tRNA, which are about 75 nucleotides, and we have 5S, which are about 120. And what we observed was that when we trained on the tRNA data, then, so that would be like a T1, then the tRNA prediction improved a lot over the standard predictions, but the 5S did not. If we trained on 5S, then the 5S predictions improved over the standard ones, but the tRNA did not. And when we trained on both of them, it improved over over standard ones, but not as much as if we had trained on the two families individually. And the conundrum was the parameters lie in very different places. So the best parameters here for tRNA have a positive initiation penalty, but I'm actually getting a bonus for every branch that I have, whereas the 5S ones are reversed. I'm actually getting a bonus as an initiation, quote unquote, penalty, but now I'm paying a penalty for every branching one. So the geometry is quite different here. And so the second most recent paper is resolving this by really understanding why you have this. I can't improve tRNA. I can improve tRNA, but sort of at the expense of 5S and vice versa. And that is because, so basically there are 50 blue trapezoids and basically 50 orange rectangles. And if you take essentially the intersection of the orange rectangles, you get this rectangle. And if you take essentially the intersection of the blue trapezoids, you get this trapezoid, and they are nowhere close to each other. So, and if you try to optimize both the orange rectangles and the blue trapezoids, you end up with this blip. And so what we ended up saying was, it makes no sense to try and come up with a single combination of better branching parameters. Because basically, if I choose, any combination in here will give me the same accuracy for the blue ones. Any combination here will give me the same accuracy for the orange ones. It's nonsense to choose this one because I grossly overfit to my training data if I do that. And so really what we should do is think about searching over a range of possible branching parameters to generate a set of possible, sample from our reparameterization options. This drove a reviewer nuts. I mean, really, it was like the review was essentially your project is morally flawed because there should be a single combination of parameters that reflects biophysical reality. And it's like, well, in a perfect world, sure, but we're dealing with a very imperfect objective function here in our optimization. And so what we're really doing is using this reparameterization. I mean, we have this like helices, loops trade-offs. And so if we remove the branching parameters from that, we're basically saying, what is the weight of the helices? What is the weight of the other loops? And then I'm using the branching parameters to kind of change the balance between these and get potentially quite different secondary structures. So the most recent results, and I won't spend any more time on this, but I'm happy to discuss it afterwards, is we came up with a way of computing the slice directly. So given an RNA structure, we can now compute with the minimum number of calls to the MFE optimization, because that's expensive, what its parameter slice looks like. And so now we find ourselves with this conundrum of I can generate a large set of possible secondary structures. How do I find the ones in this, haystack that are the most useful to report to a biologist? And so with that, I don't see any, we haven't yet found any cycles here, but, there are a lot of people at this meeting who think about different methods of trying to extract a useful signal from a noisy, complicated biological system. So I'd love to hear suggestions that you might have about what to do. Thank you for your attention.
Thank you. Any questions for Christine? Oh, there's some. Okay.
So for this needle in the haystack problem, is the kind of haystack made up of those circular, like that is the data that you have like a bunch of those?
Yes.
And some of them are good.
Right. So each colored region generates a set of generates a structure, which is a set of base pairs. And so we want to, for this particular case, we're searching in this region, but we don't actually, that region was defined by previous work. We don't know, in the current results we show, the bigger you make, I mean, again, and this is basically a tautology. The bigger you make your haystack, the more likely you are to find a needle in it. So, if I don't constrain myself to just searching within this rectangle, I will find better and more accurate predictions. If I don't constrain myself to just one slice, I will find more accurate predictions. So we can keep making the haystack bigger, but now we still don't know how to find the needle or needles in it. And a needle would be defined as a structure that is sort of likely in the sense of, so the farther away that you move, the more pathological these structures start looking. And so there is a continuum where you reach a point where you say, clearly that's nonsense. I don't have to look any further. These are the ones that are close to the default parameters generally are useful, but between those two, we don't quite understand it. But essentially each, you can think of this as like, this is kind of like a 4-color problem. And so each region corresponds to one prediction. And
just to quickly follow up on that. So the needles do you have like a set of needles that you can kind of look at topological signatures of that and then see if you can find the same signatures in the haystack? Is that your?
We do, but I don't necessarily trust them because I didn't generate the testing data. And so this is an example of where the, and the colors are not showing up particularly well. They're supposed to be gray ones and purple ones and gold ones. And so the gray are the true positives where the target or the prediction agrees with the target. And the purple are the false negatives. And so here, we're not getting this part. This is what we're supposed to get, but instead we get the gold. But then I look at this and I was like, I don't really believe those like singleton base pairs. Either we're missing base pairs or there's other things going on with the structure. And so first of all, there's a question of, as I'm sure you're aware, the testing data needs to be better curated. We probably need a different accuracy measurement because right now for convenience, it's done at a base pair level, but the base pairs are highly correlated within helices. And so if you're missing a base pair from a helix, you're almost certainly missing the whole helix. It's not clear, the structure can end up being quite modular. And so at the top, we've got two different examples of structures. And, at the bottom, this structure is closer to the target structure than that one is, but both of them if you remove the target structure, if you looked at both of them, you would not say there's anything that seems obviously, for want of a better word, like this is a biologically infeasible structure.
Thank you. So regarding this correspondence between these lattice of non-crossing partitions and these passes between trees, I was wondering, I was trying to figure out what kind of structural property of this lattice does have any good interpretation in this side. For example, looking to the chain and anti-chain, and probably looking to the Hilbert's theorem, which decomposes structure to the chains, and then try to, because every chain somehow could be interpreted in this space as the nice transitions between these passes between.
That's right.
Right. So if you look at, for example, through the lens of this kind of decompositions in this lattice, try to decompose that in a nice way, and then try to go back to the initial space to somehow move between these different passes and try to maximize, your maximization along the passes, and or even more beside this Dilworth and decomposition, there are a lot of features, lattice theoretic features in the second space, which could have potentially some interpretation in the initial space. I was wondering if you could somehow give any comments on that. Yeah?
Yes, I'm not I have not thought recently, that it is certainly likely that the combinatorics will tell us more about the structure of this or potential structure of this space. So the lattice of non-crossing partitions is fairly well characterized. But there's some interesting. So for instance, in the lattice of non-crossing partitions, there is no symmetry between how the lower bound is defined and the upper bound is defined. So the lower bound is exactly the set intersection, but the upper bound has to be the union, has basically this union of subsets. But if we think about it in the tree context, there is a very natural duality between taking the tree partition between one star tree versus another star tree. And so I think it would be fruitful to think about, to revisit some of the results on the lattice of non-crossing partitions, and to see, like, does this tree interpretation provide new insights, and also what might we learn from known results on the lattice that would help us better understand the structure of the tree configurations.
Right. Any other questions? Not, let's thank Christine
Thank
again.
you.